everyone, welcome to BioMixUp. Today in this video, we will be discussing another one topic, that is a chip assay. It is also known as chromatin immunoprecipitation assay. So what is a chromatin? When the DNA and proteins are combiningly present, they are known as chromatin. Immunoprecipitation, immuno means antigen or antibody. It may be a antigen or it may be a antibody. Next comes the precipitation. This precipitation means by the process of centrifugation or by using some of the magnetic field, we can separate the two substances. That means a soluble substances as well as a insoluble substances. Whenever they are present in a solution, they are combiningly present, but by using the centrifugation technique, we can form the precipitation. That means the soluble substances will be separated and the insoluble substances will be settled down. So by using this type of assay, like the chip assay, we can study the interaction between the DNA and the protein. In our previous video, we have already discussed some other techniques which are required for studying the interaction between the DNA and the protein. Like we have already discussed about the IMSA as well as DNA footprinting. So what happens in case a cell, here we are having a cell, and inside the cell there is a nucleus present, and inside the nucleus we can get a DNA, and inside and with this DNA there are various types of proteins interacting to regulate the work of this DNA. Suppose this is a DNA and we have now identified a novel protein, and we want to study that whether the protein is interacting with the DNA or not, and if interacting, in which particular sequence of the DNA, what is the form of the DNA, how the sequence are arranged. We have to identify that. For such kind of techniques, we can use the chip assay. So there are various steps to perform this kind of assay. So let us see. So the first step here is in vitro cross-linking of the cell. Second step is chromatin extraction. Third step is DNA searing. Fourth is DNA protein immunoprecipitation by the involvement of antibody against the target protein. That means whatever protein we have got, we know that what kind of protein is it. Against that, we can design some of the antibody and we can form the precipitation technique. The fifth step is cross-linking reversal. Sixth is DNA purification, and seventh is the downstream analysis by using the PCR or NGS, that is next generation sequencing. Suppose we know that this sequence is responsible, this part of the DNA is interacting with the protein. Now we have to find out that what is the sequence of the DNA. So for that we perform the NGS. So let us see one by one details about this steps. So the first step here is cross-linking. What happens whenever we are seeing a DNA? In this DNA, there are various types of proteins linking. This linking or the interaction may be a strong interaction or it may be a weak interaction. So whenever we are doing the lysis or we are extracting the DNA from the nucleus, if there would be a weak interaction between the DNA and the protein, it can disrupt, or the bonding will be removed between the DNA and the RNA. So for preventing that, we are doing the cross-linking. How we are doing it? We are doing it by using the formaldehyde. Formaldehyde. What this formaldehyde does, it helps in the strong binding of the DNA as well as the protein by increasing the covalent bond. So by the use of formaldehyde, we can create a strong covalent bond between the DNA as well as the protein. So now next comes the second step. So what was our second step? The second step is the cell lysis. Now we have already bonded the DNA as well as the protein in a strong manner by increasing its covalent bond by using formaldehyde. Now we have to extract the DNA from the nucleus, that is known as cell lysis. For the cell lysis, we can use some of the detergent-based solution. What is the work of this detergent-based solution? It will help in disrupting the cell membrane. So we can get the DNA extracted from it. This was the first step. You can see that it is the first step. This is the first step in which we are doing the cross-linking. That means we are using the formaldehyde for strong interaction between the DNA and the protein. The next step is that we are doing the cell lysis. That means we are taking out the DNA. We are taking out the DNA from the nucleus. Now comes the third step. So what is the third step? The third step was the chromatin preparation. So what is this chromatin preparation? Whenever we are taking the DNA from the nucleus or from the cell, the DNA is having a large copy. The strand of the DNA is very large. So we cannot study this large fragments for this assay. So for that reason, what we have to do is we have to cleave this DNA into small fragments. The small fragments can be up to 200 to 500 base pair. Okay. So how can we do this? We can do this by using the sonication as well as by using some of the enzyme like DNase 1. So already in the last video, we have discussed about this DNase1 in the video of DNA footprinting. What was the work of the DNase1? This DNase1, it is a endonuclease and it cleaves the DNA in between, but not in the terminal end. And already we have discussed that in the protein, in the DNA, if it is interacting with this protein, if this part is interacting with the protein, the DNase will cleave only in this part, but it will not cleave in that area in which the DNA is interacting with the protein. By the use of sonication as well as by using certain enzyme like DNase1, we can do the chromatin preparation. So whenever we are treating it with the enzyme or doing sonication, then we will get the fragments. So here are the fragments. In the second part, we can see that here there are fragments where only the DNA is present, and here we can see that the DNA are interacting with some of the proteins. So now we got the small fragments. So what next? In this fragment, in this fragments, here the proteins are interacting. This we cannot see during our experiment. We cannot see that because we are doing everything in a solution. So the next step is immunoprecipitation. What happens? We are having a DNA and this DNA is interacting with some of the proteins, and we know this what kind of protein is it. Now this protein can act as an antigen. So now we can design an antibody specific to this antigen. That means the antibody can specifically bind to this antigen. So in a solution, we can add the DNA, like we can see here. Here there is a DNA as well as the proteins which are interacting with it. Not only the DNA and the protein which are interacting, all the DNAs will be present, the DNAs which are not interacting with the protein as well as the DNAs which are interacting with the proteins. Now here we will add the antibodies which we have designed specific to the antigen. So now there will be a immunoprecipitation. What do we mean by immunoprecipitation? That means we are doing a centrifugation in which the particles which are insoluble, or we can also tell that they are having high molecular weight as compared to the DNA. If this DNA is interacting with the protein and the protein is interacting with the antibody, it will obviously have high molecular weight. That means it will settle down when we do the centrifugation. In this way, here there is a settlement of the proteins, DNA as well as proteins in the bottom, and the rest of the things will be washed off here. These are only the DNAs which are present, which is not binding with the protein, because this is not binding with the antibody. If it would bind with the protein and antibody would come and bind that, it will be settled down. So these things needs to be removed. Here we can get that DNA which is binding with the protein and the protein is linked with the antibody. So whenever we are designing a antibody, in that we use the magnetic beads. Magnetic beads. If we are using the magnetic beads in the antibody, it will either during the process of centrifugation it will be settled down, or else we can separate them by using magnetic strip, or we can separate them by using the magnet. Or else what we can do, we can tag this with biotin. And in this tube it will, and if we are using the biotin in the antibody, the tube which we are taking, it should be coated with avidin. So if that is binding with the avidin and the antibody, it will be settled down. So here we can see that by the process of immunoprecipitation, the target protein and the DNA is bounded to the antibody and they are settled down. Now what next? The next is the cross-linking reversal and DNA quantification. So for the strong interaction between the DNA and the protein, in the first step we have used the formaldehyde. Now, what we want to know, that if the protein is interacting with the sequence of the DNA, in which particular area it is coming and interacting. We have to know that. For that, what we have to do? We have to do the sequencing. So, through the precipitation, what we got the DNA. We got the DNA, along with it is binding with the protein, and this protein is being interacted with this antibody. We have to remove all this. We can remove this protein as well as antibody by treating it with an enzyme which is known as proteinase K. This is an enzyme which can remove the proteins from the DNA. Now what we got? We have only got the DNA which was binding with this protein. So now we can give this DNA for PCR for amplifying this. Next, we can give this for NGS sequencing. Whatever kind of sequencing we want to give, we can give it. By the process of sequencing, we can get in which sequence the protein was interacting with the DNA. The next step is the DNA quantification. The last step was the DNA quantification. For DNA quantification, we will get the specific DNA. After treating with proteinase K, we will remove the protein and we will give only the DNA for PCR, QRT PCR, or sequencing as well as microarray for further analysis. By the using this diagram, let us overall see how this process was done. So we was having a cell. Inside the cell there is a nucleus, and inside the nucleus there is a DNA, and this DNA is interacting with some of the proteins. For the strong binding, first what we did is we did the cross-linking by using the formaldehyde. This formaldehyde covalently helps in the covalent interaction of the DNA as well as the protein. The next step what we did is we did the cell lysis, by which we brought the DNA outside from the cell. The next step here what we got a long stretch of DNA. So for doing this assay, we cannot do this with this long stretch. We require small pieces of DNA. So by using the sonication or by using certain enzymes like DNase 1, we cleave this DNA into small fragments. These small fragments can be of 200 to 500 base pair. As we already discussed in our previous video, the work of DNase 1, this DNase 1, it is an endonuclease and it can cleave the DNA from the middle. And the part which is interacting, in which the DNA and the protein are interacting, it cannot cleave that area. So here what we got the DNA, only the DNA may be present here, as well as the DNA and the proteins which are interacting, they must be present here, which is of small size, that is the 200 to 500 base pair. The proteins which are interacting, we have already known which kind of protein we want to study. So against them we have designed the antibody. Now we have added the antibody and we have did the immunoprecipitation. That means by using the antibody, we have did the precipitation by using some of the centrifugation step, in which the substances settle down and we have cleaned other thing, or we have washed other things, the impurities or some of the DNAs which are present inside, which are not bound with the protein, they are been washed up. Now what we got, we have got only the DNA which are present. So here it was settled down and we got here the DNA, protein as well as some of the antibody. Now what we have to do, we have to remove the proteins from the DNA by using some of the enzymes like proteinase K. This proteinase K help in the removal of the protein. So now we got this sequence of the DNA in which the protein was interacting. This DNA should be for the given for analysis. That means for knowing the sequence. So for that, we can give it for sequencing. It can be any NGS sequencing or microarray PCR if we want to see how much it is expressing, if this protein is interacting with the DNA, in what amount some function is expressing. So in that case, we can give the qPCR. We can give it for qPCR. This is all about the chip assay.